Hello everyone, I'm Amaresh. So today I thought of having another episode on RNA biology. As you can see, my lab is RNA biology lab. So RNA means ribonucleic acid. Most people know RNA is transcribed from DNA that gives rise to protein, and that is called the central dogma. But uh, interestingly, most RNA do not code for proteins. So majority of the RNA transcribed from the human genome are non-protein coding. So today we'll be discussing what are different kinds of RNA, how they are involved in either making the protein or they regulate the protein synthesis process. So let's discuss how many types of RNA are there and what they do in a cell. So as you all know, DNA is the life molecule that gives rise to RNA and then that gives rise to protein. So this is called the central dogma. But uh, this all the DNA in the cell doesn't transcribe into RNA that produce protein. So if we look at the human genome, it is a highly condensed DNA molecule. They are packaged into chromatin and we have 23 pairs of chromosome with a very dense DNA content. So these are like coiled, supercoiled DNA present in the chromosome. So, and if we think of the length, uh, then we have like 3.2 billion bases per cell. If we isolate that, it will be like 2 meters long. It's very huge from individual cell. And if you isolate all the DNA from any single individual of any of us, it will be like 6.2 billion kilometers, which is huge. And to know like how long this could be, let's see the distance between sun and earth. It's only 150 million kilometers. Then we can go and come back from sun 41 times if we make a rope with all the DNA isolated from any single individual. So that big DNA we have in our in and the chromosomes. So if the RNA is so huge, so they are transcribed into RNA. As you can see here, they are mostly showing here mRNA, and that is the protein that most people believe this is how genes are expressed. But if we look at the history, people have discovered ribosomal RNA even before mRNA. And then till today, in last 6-7 decades, we have discovered mRNA that translates into protein. There are catalytic RNAs. There are H19 GIST, which are long non-coding RNAs. In 1990s, we have discovered microRNA and so many different kind of RNA with the advancement of next generation sequencing and novel pipelines for the analysis. We have discovered tons of different types of RNA and different number of RNA molecules, and the most recent one is the RNA circles that my lab works on. So that was in 2011 and 12. RNA circles were discovered in many types of cells through RNA sequencing. So if we look at the human genome, so most of the genome, like 76% of the genome out of 2 meters, like you can imagine 1.5 meter is transcribed into RNA. And if you take the whole pool of RNA into consideration, only 2% of them is coding for protein. Others are non-coding RNAs. The small non-coding RNAs are less than 200 nucleotides. They can be snRNAs, snRNAs, or they can be microRNAs. Okay, and then the long non-coding RNA is intergenic RNA, antisense RNA, sense RNA, and linear long RNAs. They do not code for protein. Even the RNA circles are circular RNA. So in a cell, if we isolate all the RNA from any cell and then want to see by mass which one is the most abundant one, what we will see is rRNA is the most abundant one, and then followed by tRNA and mRNA. And if we run them on a gel, what you will see is all the rRNA, 28S, 18S, and 5S. And this bottom band is not only 5S RNA, it can have tRNA, snRNA, and other small RNA population here. Although by mass you see mRNA percentage is not that bad, so we probably should see them on jet. We don't see them because there is not a specific length for mRNA. They can be like 500 bases, they can be 5 kilobases, or even 10 kilobases. So the length varies. So that's why they are present throughout the gel. So we cannot see mRNA. So many times students answer, oh, if we run the RNA on gel, will see mRNA. No, you cannot see mRNA. So mRNA are there, but you cannot see them. All you see is your 28S, 18S, and 5S rRNA, and the tRNA and snRNA probably you can see, not the mRNA or the microRNA also you cannot see. Okay, so the length varies. So that's why they are present throughout the gel. So we cannot see mRNA. So many times student answer, and by number, if we look at, so the majority of the RNA by number in any cell is tRNA. Then your rRNA followed by mRNA, snRNA, link RNA, and other microRNAs. But tRNA by number is the most abundant RNA type in any cell by number. But by mass, it is always rRNA. So coming to the function, as we all believe, like a DNA transcribes into messenger RNA that codes for the protein. So mRNA that codes for protein, but rRNA and tRNA are integral parts of the translation machinery. Without them, translation cannot happen. And then we have snRNA, small RNA. These are involved in processing modifications and mRNA splicing. Without snRNA, our splicing cannot happen, so we cannot have mRNA for translation. And then finally, the regulatory RNAs. So these are RNAs which regulate which gene has to be expressed at which stage and how much they need to be expressed. So they basically determine the quality and quantity of gene to be expressed in any cell, any type of tissue. So based on their function, so we can categorize them into four different types. One codes for the genomic information, that's mRNA. Then RNA also makes structure. For the ribosome structure is mostly determined by the rRNA. So RNA can give structure to a organelle or biomolecule, and then it can be catalytic. So introne splicing is one of the best example. So group 1 intrones, there are self-splicing intrones. So those are RNA, but they catalyze. So those are called ribozymes. And then the regulatory ones, as I said, like microRNA, link RNA, and circular RNA, they can regulate the gene expression. Like one of the RNA, regulatory RNA, is microRNA. It's a small RNA of 19 to 21 nucleotide long. So, and they mostly regulate mRNA stability and translation. So let's see what are the functions it has in a cell. So This microRNA mostly targets the 3' UTR of mRNA and inhibit the cap mediated translation by inhibiting the cap binding proteins. And also, it can activate the degradation of RNA by activating the endonuclease. And if it has a perfect complementarity, the mRNA gets degraded. Then the other thing it does is inhibit the most of the RNA like to be translated when they are in a circle, and this PABP interact with the Fourier complex and the cap binding complex, and they get translated efficiently. But this microRNA inhibits this thing, so it reduces the translation. And then the second thing it does is inhibition of the ribosomal subunit. So it can also inhibit the ribosome binding and moving along the mRNA. Then the other thing microRNA can do is premature ribosome drop off. Then the other thing, another possibility is that it can slow down the translation efficiency, and also it can degrade the protein itself. Nascent protein can be degraded, and finally, this microRNA binding to mRNA, they can induce the mRNA to go to P bodies and stay there without getting translated into protein. So this is a very small RNA, but it has lot of role in regulating the gene expression. Another regulatory RNA is a link RNA that is transcribed as mRNA by Pol2 from the genome, but it doesn't code for protein. So it can inhibit or regulate the mRNA stability and translation by directly interacting with mRNA. It can act as a competing endogenous RNA for the microRNA, so that the microRNA doesn't bind to the target mRNA, which is supposed to inhibit the mRNA translation or induce degradation. Now the microRNA are bound with the link RNA. So basically, link RNAs can act as sponge for the microRNA. They can also interact with the chromatin and regulate gene transcription. They can also activate transcription. They suppress and can act as activators. So from transcription to translation and mRNA stability, link RNA regulate most steps of gene expression. The last and the most interesting regulatory RNA in the RNA family is circular RNAs or RNA circles. These are closed loop RNA molecules without any 5' or 3' end. So they were discovered in 1980s by electron microscopy, but most people thought these are splicing errors, or probably these are endonucleases. Of course, they will be endonucleases, but no one thought these are genuine RNA circles, not the lariates. Uh, so nobody basically paid attention to these kind of novel RNA molecules for almost 30 years. Although there were articles talking about RNA circles, and people also found exons like scrambled exons, like exon 2 coming after exon 3, but they thought, okay, this could be splicing error, so there is a reshuffling of exons happening. But the whole field missed the point that these scrambled exon could be actually RNA circles. But in 2012 and 13, there were many articles. Uh, they found that these RNA molecules, the RNA circles, are present in all kind of eukaryotic cells, and the number is huge. And after the first discovery from NGS analysis as well as biochemical validation, a lot of people in the field jumped into circRNA research. And now we have thousands of articles on RNA circles, and they all say the same thing. Like these are ubiquitously expressed RNA molecules from the pre-mRNA. So if you look at the pre-mRNA, the canonical splicing gives rise to the mRNA. But there are possibility of non-canonical splicing, which is like back splicing. The end of any exon attacks the start of the same exon or start of the upstream exon. So that way it makes circles from the exons. Sometimes the introns are included, so those are called EI circRNAs, exon intron circRNAs, or there are splicing lariats, which are stable lariats. They are not degraded by the differentiating enzyme, and they are called as circular intronic RNAs or CIRNAs, or intronic circRNAs, or some people name it as SISRNAs. So all these RNA molecules are circular in nature. They do not have free ends for the exonuclease to work. So how we detect them? These novel RNA molecules can be detected by PCR, that is the most used method. So in PCR, we always put two primers facing towards each other. But in case of circular RNA, we can put two primers facing outwards. If the primers face outwards, in case if it is actually a scrambled exon and it is not making a circle, then the two primers will never meet and give you a PCR product. But if it is a circular RNA, the two primers will come together to make a PCR product. And that is the most used method and the convenient way to find if one RNA is actually a circle or it's a linear one. And there are other methods by the northern blotting, 2D gel electrophoresis, or gel trap. uh, these are like not much used for circRNA detection, but those can be used. And the other most relevant method for isolation or identification of circular RNA is by exonuclease treatment. So if we treat the total RNA with any exonuclease, suppose RNHR, it's a 3' to 5' exonuclease, so you will degrade the linear RNA. Not all linear RNA will be degraded, but most of the linear RNA will be degraded, and leaving you with an enriched population of RNA with RNA circles. And these enriched population of RNA circles can be sequenced in RNA sequencing to find the uh, circular RNA. So if you know how RNA sequencing works, you must be knowing that RNAs are fragmented. So let's see here. So you have the genome that is transcribed into pre-mRNA, then you have the mature mRNA after canonical splicing. From canonical splicing, you fragment them into multiple fragments, and those fragments can be added with adapters for sequencing, because adapters will help you in amplifying those fragments, and that will go into the sequencing machine. And the sequencing machine have millions of binding spots. Basically, those are the million of reads that can be read at a single time point. So in suppose we have Novaseq, so you can read in a flow cell 800 million RNA fragments at a given point of time. So you have tons of fragments, those are having adapters which are amplified, then put it for the sequencing, and then in the sequencer you get the sequence traces. And those sequence traces can be aligned with the genome or the transcriptome to find out which of the RNA molecules are expressed in that cell type, because genome is same in all cell types, but the RNA they express or the RNA type or the splice variant they express are different in different cell types that make them unique. So with the alignment, you can figure out which RNA are expressed in the specific RNA sample you have sequenced. This is true for the linear RNA, but if you want to go and find out circular RNA from any RNA sequencing data, it is tough because majority of the reads will match with the linear transcriptome or the genome, but there are fragments which doesn't match, like maybe you can say 5 to 10% of the reads will not match with the linear transcriptome. So those are called fusion reads. Those can be used to find out if those fusion reads are coming from RNA circles. So how? Let's understand this. So in linear RNAs, the linear read perfectly align with the transcriptome or the genome. But in case of circular RNA, the last half of the RNA-seq read actually aligns with a upstream sequence and the first half aligns with a downstream sequence. That's the first principle how the RNA secrets are aligned. Once this alignment is done, the program uh, find out where is the break point, where the read is breaking into different halves. And those breakpoints on the transcriptome is taken as the end or the start of the circular RNA, and the program reconstruct a circular RNA by combining all the exons present between the 2 points. Those are matching with the RNA secret. So that way you get a circular RNA, suppose starting from here to here. And these 3 exons are included in the RNA circle. So this is not very complicated for some of you. So let me make it a little bit simpler. So if you say Brick Institute of Life Sciences Bhubaneswar as your transcript of interest, or this is your transcriptome sequence, and we are going to do an RNA sequencing. And we fragment this transcript into multiple fragments. They can break at any of the points. You add adapters, go for sequencing, and then align that with the primary transcript sequence we have, that is Brick Institute of Life Sciences Bhubaneswar, and now the reads will align like this. This is how the program works. But there could be fragments that read Sciences Institute and Sciences Institute. So the first half of the read matches here, and the second half of the read matches upstream. So this is only possible if we have a RNA circle that starts from institute and ends at sciences. So this is how the program detects where the read is breaking and how it is aligning with the reference transcript. With that, now you have a RNA circle that is read as Institute of Life Sciences, and this can be read from any start point. You can read from Science Institute of Life, or Life Science Institute of, or of Life Sciences Institute. So depending on where you are starting, you can read it, but the meaning will be different. So the same way, if any RNA is made into a circle, the meaning will be different because there is no start end. So in the sequence, the meaning will depend on where you are starting to read the sequence. So circular RNA can have multiple functions because of that. And as of now, like this is a data from 2020, but the numbers are much more now. You can see in humans, you have more than 1 million circular RNAs. And also in other species, we have tons of other circular RNAs discovered. So this tells us that the circular RNA numbers are huge and they are very abundant, ubiquitously expressed, and many of them are actually conserved and they show tissue specific expression. Then what these circular RNAs are doing in a cell. So the first thing they do is act as a microRNA sponge. They bind to proteins to act as protein decoy. They can act as transcription factors, bind to transcription factor, regulate transcription. Some of them are also secreted into the body fluid and they act as biomarkers. And recently, many circRNAs have been reported to be translated into proteins. So, the non-coding circRNAs are now moving, some of them are moving into the coding RNA because they can bind to the ribosome through internal ribosome entry site and getting translated into polypeptides. Okay, so what my lab does. So, as I have said, my lab works on circular RNAs. So, one of the system my lab focuses on is skeletal muscle. So, skeletal muscle is required for body posture, our all kind of movement, breathing, even thermogenesis, maintaining the body temperature. All these things are maintained by the skeletal muscle. And skeletal muscle is basically the most abundant tissue in the human body. It has like 50% of the protein and it constitutes 40% of the body weight of any human being. And these muscles are formed by a process called myogenesis. This process starts from the satellite cells present in the muscle. These are quiescent stem cells. They differentiate into myoblasts. These myoblasts then start forming elongated myocytes, and these myocytes then form long multinucleated myotubes. But this whole process is controlled by many transcription factors. They are expressed at a specific point of this whole process and regulate the myogenesis. But these transcription factors are also regulated by many non-coding RNAs from link RNA to microRNA and circular RNA. So, so if we look at, so this whole process is regulated by transcription factors, microRNAs, link RNAs, and RNA binding proteins. So my lab mostly focuses on how RNA circles or circular RNAs regulate myogenesis. That's the my lab focus. And one of the recent article we have worked on is circular NFIX that act as a decoy or sponge for microRNA 204, which is known to act on MEF2C mRNA. And with increased circNFIX, the microRNA 204 is no more available to inhibit MEF2C. So MEF2C is getting translated more or expressed more. So that leads to myogenesis. So that is one of the recent paper from our group. And we are also working on other aspects of circular RNA and how they regulate myogenesis. And thanks to the funding agencies, the major funding for my lab came from Wellcome Trust DBT India Alliance for the last 5 years, then SERV, DBT, and of course Institute of Life Sciences. That's my host institute for all the intramural support and the infrastructure. And lastly, but not the least, my group. So this is the team that works on these novel RNA molecules and discover their novel functions. And if you want to know more about the research we are doing, our other activities, please go to our website and find out more. And if you have any more questions on RNA or the work we are doing, please follow us, and you can write email to know more about our research activities and outreach activities. So follow our channel or Institute of Life Sciences channel to know more about the research and other activities we are doing. Thank you very much. Thank you.